if you take a stroll through the lush green forests of Brazil, you will expect to find a lot of different species of animals. Colorful, different sizes, loud. The problem is that you will be looking for one particular, really native type of primate in there, and it will be either difficult or completely impossible to find the golden lion tamarin. The golden lion tamarins are an endangered species to Brazil, and there are about 3,000 of individuals left in the world, and there have been there have been really rigorous conservation efforts going on for about 50 years now, because in 1970s there were about 200 of them left in the world, both in captivity and in the wild. And so we kind of put them up into that 3,000 individuals number, but that is still not enough to sustain that population in the wild. And 50 years is quite a lot to be working on something like that and still not succeed completely. And so we decided to help from our laboratory from Stout. And the way we wanted to help is to find the tamarins, or the individuals that would be the furthest apart in their genetic composition from each other, so that we would be mating individuals who are the most not closely related. And the reason for that would be that we want that variation in their genomes for the strengths against the common diseases, and to combat that founder effect that we like to call when we have not enough individuals in the wild. So what we can do is we can take the genome of one tamarin, and then compare it to the genome of another tamarin, and then see how closely related they are. Well, how do we do that? Well, we acquired a sample of liver of one tamarins from Omaha Zoo, and we basically sequenced the genome from that tamarin. We took the DNA out of the cells, and then we found the highly variable parts of the DNA. So we call them microsatellites, and really what that means is that it's a bunch of repeats of nucleotides, or the building blocks in the DNA. And then we can compare about how many repeats we have in different individuals. So we sequenced that tamarin, and we covered about 96% of the genome. Interestingly enough, when we were starting, it was the first genome ever sequenced for tamarins, and we were kind of going hand in hand with other researchers. So now we have that sequenced assembly, and we designed primers to actually amplify those regions, and we can really just test tamarins in the zoos and find the most suitable mates for each individual, and then determine how much we want them to mate. And so we can together now save golden lion tamarins one nucleotide at a time.
Once they're mated, are there ways to get them back into the wild? Is that part of kind of populating, in Brazil once, these are created?
Yes, it's an excellent question. Thank you. So ultimately, it is the goal for the whole sequencing part. But like I said, there are only 3,000 of them left, and so we can't really find that. It will take us a really long time to kind of get to that point, but ultimately we would want to spend, maybe a few years to get to that point, because now with the sequencing that we can do, it will take us about an evening in the lab to sequence the whole genome, really. So it shouldn't take long from now to that point.
So you mentioned another research team as you were speaking. So what sort of have you engaged in collaboration with other research teams? Is that was that part of your process?
No, we actually didn't. We just found out that they were doing the same kind of research, and life happens. Sometimes that happens. They saw that it has never been sequenced, the golden lion tamarin specifically, and they decided to sequence them too. The focus of their study was a little bit different. They were not looking for microsatellites and things like that, and they were sequencing other individuals. So we solely focused on the tamarin and designing those primers so that we can really commercially give those primers out to the zoos.
Is this a method of recovery that's been used for other species?
Yes. So the method of recovery that we used was the long nucleotide, long DNA strands, and I'm not sure if it's commonly used the genome sequencing for these purposes. But the thing is, it should be because it is so easy and fast and even cheap. The machine that we have here at Stout costs only $1,000, and it looks like a thumb drive rather than, a high throughput sequencer. So it's something that zoos can do on their own, but I'm not sure if they have been. So we need to spread the word that it is possible.